All right, we'll get started. This is a very exciting research conference for me and for the DCRI because I think we're gonna do two things today. One is welcome back David, who has been over here before to talk about his research, and obviously a lot of that research involves collaborations with faculty here at DCRI. But the other part of the excitement is we've actually co-opted David and his team into starting a new Center for Discovery Genetics here at the DCRI. And so in conjunction with today's research conference, there's a little brochure that's around, but what we're going to do now is launch a formal collaboration between David's group and the clinical trials group here at DCRI to really give us a capacity to look at the pharmacogenomics of the therapies that we're working on in a way that it's never been possible before. It's never been possible because the science has moved, but it's also never been possible because we've never set up a formal collaboration like this. We really think this is going to be, this collaboration, and I was just talking with a friend of mine in the back. I mean, I think this is really going to be unique and novel to marry kind of the capabilities of the DCRI and the capabilities that you're going to hear about from David's lab and David's group. So let me introduce David. David's a geneticist that got his PhD at Stanford, spent a little stint in London, and then has spent the last 5 years here frequenting the coffee shops of both Durham and Chapel Hill. And in that time, his work has really progressed from kind of looking at core pharmacogenomic questions to looking deeper and asking questions. Why is the technology that we have not finding all the things we would hope to find in 2000? You know, Francis Collins, now director of the NIH, said in 2000 that we're going to find all kinds of stuff as a result of the $15 billion he spent on the Human Genome Project. And David pointed out both in the New England Journal and New York Times that really has been a bust up till now. So I think one of the exciting things about today's conference is that the group has actually answered that question in terms of new research hypotheses about how to use these tools to address the issues to human health that we've all are so interested in. So David's been instrumental in the lab and working with John McCutcheon and IL-28 and hepatitis C, also on the drug safety side, and looking at adverse events that are genomically based. So, David, welcome formally to the DCRI, and look forward to this conference today.
Well, thank you very much, Kevin. It really is a great pleasure for me to be here presenting what will be my first presentation at the DCRI as a regular faculty member of the DCRI. And I would like to say that that is a real honor for me. I have long been a real admirer of the DCRI's expertise in clinical research. And I really do think that we really do have a very particular opportunity here at Duke in bringing together the kind of high throughput genomic analyses and data analyses that we've been developing in the center with the diversity of the clinical expertise that's represented here at the DCRI. So what I'd like to do today is tell you a little bit about some projects that we've been doing so far, but also really telegraph where the technology is going, and I hope to use that telegraphing of the technology to plant some ideas and some seeds here about what kinds of things might be possible in the years ahead. And I would say that that also might include improving my relationship with Francis Collins, because I do think that we finally probably are at the point where we really can do a lot of directly clinically relevant work with genetics. It is true that I don't think we've done that much so far for common diseases, but I do think that's going to change, and I hope I'll get that orientation across here. So what I'd like to do is just start out by telling you a little bit about the center that was recently established at Duke. The overriding mission of the Center for Human Genome Variation, established earlier this year, is to identify clinically relevant and biologically informative genetic variation related to human disease predisposition and treatment response. And within the confines of the center, I would say that we really do have the expertise on the side of data generation and on the side of statistical, genetic, and biological interpretation to a degree, but where we really require the partnerships is on the clinical side. This is the faculty structure for the center. I direct the center. Kevin Cheyenne is the director of operations in the center, and he is here. And then there are 4 faculty members that largely concentrate in specific areas within the center that are indicated here. Anybody that's interested in collaborations with the center can make contacts really first at any one of these points, either Kevin or me, or in specific areas, any of the faculty members. All of the faculty members that are pictured here are present except for Erin, who is at a parent's day with her son. So I want to first just give you an idea that clinically relevant findings really are being made using contemporary genetic and genomic technologies. Here's a few examples from our own work. There are, of course, other examples in the field as a whole. In 2007, we used a genome-wide association approach. I'll tell you a little bit more about that in a minute, to identify gene variants that are associated with the control of HIV-1. In 2009, I'll tell you a lot more about work that we did connecting genetic variation to the therapeutic outcome for treatment of chronic hepatitis C infection. Using the same genetic data, we also looked at a leading adverse event and identified a strong predictor there. And we have also made some progress in what I consider a much harder area, which is not just drug responses, but also predisposition and neuropsychiatric disease. Here, we have been able to make connections between rare structural variation in the genome and risk of both epilepsy and schizophrenia. I think that the kinds of approaches that I'm talking about will be paying dividends in disease predisposition studies that have clinical meaning, though I do think that will be over a longer time course than in many areas of pharmacogenetics. We also recently had a discovery where we were able to identify the genetic basis of a Mendelian disease by whole genome sequencing, and I'll give you an idea of how that worked. So those are just some highlights. I now want to try to flesh in a little bit of details in some of those areas and then try to project where we're headed. So I first want to tell you a little bit of the background, which will be familiar to many people, of the story for hepatitis C, but also fill in a little bit of new information of where the story has got to most recently. And the work that I'm going to talk about first came out in these 2 papers in Nature, and I just want to use this to emphasize that this was a very broad collaboration that led to this work. The primary collaborator for all of the work all the way through was John McCutcheon, right from the initiation and conception of the studies and discussions with Sharon Plow. And we have also had a number of other individuals involved increasingly over time, including Andrew Muir, who is here today, and then also folks from the center, Jacques Fillet, Dongliang Ge, who have been doing a lot of the analyses. And then all of the data generation has come in the usual way from Kevin's lab. And Tom Urban, who is here, has been involved in a lot of the attempts to work out the biological bases of these associations. So just to set the stage for this work, it has been known for a long time that treatment for chronic infection with hepatitis C is an area where you would like to better understand variable responses to treatment. And that is both in terms of efficacy, but also in terms of adverse events. The reason for that is that the treatment does not always work. In fact, it probably only works about half of the time. And the treatment is always a pretty arduous affair. It goes on for a long time, up to 48 weeks, and it's associated with unpleasant and sometimes dangerous side effects. And for those reasons, you'd really like to understand something about who is likely to be cured by this treatment and who's likely to suffer different adverse events. Some details about that are indicated here, but overall, in individuals of European ancestry, patients are cured only about half of the time, and there are a series of well-known treatment-limiting adverse events, including anemia. So we decided to make a genetic investigation of responses to treatment using the samples that emerged from one big clinical trial, which was called the IDEAL trial. The trial was not set up in order to do genetics. It was set up to compare different formulations of the standard of care treatment which combines an interferon alpha formulation along with ribavirin. And this trial compared different formulations of interferon alpha. But they had really good response data and also really good information about the adverse events that were observed. And because they thought it might be interesting to look at genetics, they also had blood samples. So we were able to put together a collaboration to access just over 1,500 of the samples and carry out a genome-wide association study and look at both treatment outcome and also adverse events. And let me just say now a word about how genome-wide association studies work. What you do in one of those studies is you get a commercial chip that has on it sufficient reagents to interrogate somewhere now between 500,000 and 2 million common, usually common, single nucleotide polymorphisms in the genome. And what those variants do is give you information about essentially all the common variants in the human genome. And so when you do one of these studies, if there's a common variant in the human genome that influences the trait, you will see evidence of it in the study. And by common, we mean that the allele frequency for the minor form is more than 4 or 5% in the population. So do one of these studies. If you have that kind of genetic control, you're likely to find the evidence for it. So that's what we did. We used chips that had just over 550,000 variants interrogated on them at that stage and looked at association with who was cured and who wasn't. The lingo in the field is to refer to that as sustained virological response. That just means you stop the treatment, you look to see if the virus is there, the virus is gone, they had SVR, they were cured. And we just asked what variants associate with who was cured and who wasn't cured. And we found that there were a set of variants in chromosome 19 that clearly had significant association when you correct for all the tests you're carrying out with who was cured. And those are the indicated variants that have association there. And I just say by way of interpretation, if I can figure out how to do it, maybe not. What you're looking at here is all the variants that were interrogated on the x-axis there lined up, and on the y-axis is the significance value. And for experiment-wide significance, the threshold is around 10 to the negative 8, so ending up there at around 8 on the log scale. And what you see for those red points is that you're at 10 to the negative 28, so clearly genome-wide significant. There's the region where the variants are found that are associated. They all cluster right around IL-28B. There's the p-value for those variants, and there's just a blow-up of the region. So what we have is a set of variants strongly associated, clearly significantly associated with who is cured and who isn't. Definitely a significant association. Not only is it clearly significant given the sample size, but the resolution it offers in terms of making predictions about what's going to happen to an individual patient is of a sufficient magnitude to be of interest to both clinicians and patients in many settings. Okay, now I've chosen that formulation very carefully. I do not mean to say that there's a clear algorithm about what you do by genotype, but I do mean to say that there's a difference amongst patients that will often be of interest to both the clinician and the patient. And here is an illustration of the difference. What I show here are individuals with the good response genotype for the variant that we first discovered associated with response, and here the poor response homozygote genotype for individuals of European ancestry, and on this axis, the proportion of individuals that are cured after they go through the course of treatment. And what you see for the good response individuals is more than 80% of them will be cured, and for the poor response genotype, it's just over 30%. Now, this is by far the strongest predictor that is known in Hep C that is pre-treatment, before you start treating individuals, before you look at what happens during the early course of treatment. And it is also a difference for many clinicians that I've spoken to that they indicate they would like to discuss with their patients. So I think we do have here a finding with clinical relevance, even though there isn't algorithmic certainty about exactly how you would use it. And as a lot of people here would imagine, that has led to a lot of long discussions between me and Rod Califf about what we ought to say in terms of clinical relevance here, given that there isn't algorithmic certainty about how it ought to be used in the clinic. The other point I want to make here that's interesting is that not only do we see a very strong effect of the variant within individuals of European ancestry. We also see that the variant has the same effect in individuals of primarily African ancestry. And what I didn't highlight on the earlier slide is that it's long been known, in fact, in work that really was initiated by Andrew here at Duke, it's long been known that individuals of primarily African ancestry are much less likely to be cured by this treatment than individuals of primarily European ancestry. And what we see is that the variant has the same effect in its direction in individuals of primarily African and European ancestry, but the variant that leads to the good response is less common in African populations than European populations. And in fact, this variant, therefore, is responsible for more than half of the well-known difference in response rates between individuals of European and African ancestors. So we actually explain that, most of that difference with this finding. So where are we now? There is a set of variants that have been, in different studies, associated with response. This is the variant near IL-28B that we first associated, and there's a whole bunch of other variants that have been associated. We now know that it is unequivocally a real finding. It has been replicated in probably more than 15 published studies so far. Of course, we knew it was real already because when you do a genome-wide association study correctly, you know how to interpret a clear finding. But now there is clear confirmation in the field, so there's no question this is a real finding. And I should also make one other point. For those that sort of keep track of what comes out of operations, not that much comes out of operation. Let's be clear about that. So we've been working for 5 years now in the center, and we have a few discoveries. Okay, so I will say not that much comes out. But what I will also say is what does come out looks like this, and that's what's been the case so far, and that's how we intend to keep it. When we report a discovery, then other people look at it and they find the same thing, and that really is something that we try to specialize in, making sure that we really only make claims that can really stand up. This has unequivocally been replicated, and the only other comment I want to make is that the set of variants that have been associated all have similar evidence of association in individuals of European ancestry to this one. But if you look at individuals of African ancestry, this one, in fact, is stronger than the others, and so that is the one that we consider to be the best choice as a diagnostic. The other comment I want to make is not only has this now been replicated well, and so we know it's a real finding, but perhaps more importantly, there is clear clinical interest in using this. So the test is now offered by LabCorp, and I don't know actually how much it's being used right now. I don't know if we can actually work that out, but I do know that many clinicians that I've spoken to have ordered this in their clinic. So that's the sort of good news for the story. It's definitely a real effect. It's definitely being used in the clinic. The sort of less inspiring part of the story right now is that we really don't have a clue how this works, okay? So we have a set of variants that are near IL-28B. They very clearly associate with what happens following treatment for chronic hep C infection. The gene I'm now going to tell you a little tiny bit about. The gene encodes a interferon lambda, which is, of course, related to interferon alpha, which is what's used for treatment. It's known that interferon lambda goes through a different receptor complex to stimulate interferon-stimulated genes, and it's thought that even though it goes through a receptor complex, the downstream signaling pathway is the same between alpha and lambda. And so that's what we know about the gene where the variants are found, and what we wanted to do is ask whether we were able to work out why genetic variation near this gene might influence response to treatment. And I should also say we know that it also influences spontaneous clearance of hepatitis C. Now, we've done a few things to try to address that. We've asked whether the variants influence the expression level of the gene product, which then might influence the amount of signaling and somehow have an effect on response. We have also asked in an in vitro system whether an amino acid replacement variant associated with a discovery variant has different potency against a hep C replicon system in vitro. Those studies were completely negative. We didn't show any effect there, so again, we don't have the slightest idea what's happening. And the only thing we've been able to see so far that gives us perhaps a hint at what might be happening is that when we look at the expression levels of genes in liver biopsy samples from individuals before they go on to treatment to look at the baseline expression, what we find is that there is a clear effect of the IL-28B genotype status on the expression levels of interferon-stimulated genes. And in particular, what we see is that individuals that have the bad response genotype have higher basal levels for interferon-stimulated genes than individuals that have the good response genotype. And so from that, Tom Urban, I think, was the first to formulate the idea that what's happening is that maybe IL-28B genotype can lead to an over or inappropriate interferon response, which then kind of wears out the system, making individuals less responsive when they actually are given interferon alpha. I have no idea if that hypothesis is right. If anybody wants to attack it, then they can take that up with Tom. But that right now is probably the only really credible hypothesis that's on the table. My own orientation is that perhaps the problem is in the replicon system and that it is the amino acid replacement polymorphism, and we just can't see an effect of it. Probably one of those two things is what's happening. In any event, we really don't know what's happening mechanistically right now. I now want to turn to our studies on looking at one of the leading adverse events, and that's anemia. And the majority of individuals that get standard of care treatment for chronic hep C infection suffer a significant decline in hemoglobin levels, and a minority of those individuals, a significant minority that have the decline in hemoglobin levels, have to have a change in ribavirin dose because of the effect on hemoglobin. And it's thought that ribavirin is the primary agent that's driving the effect on hemoglobin. So we also wanted to see whether genetic variation affects the change in hemoglobin. We again carried out a genome-wide association study in exactly the standard way. We used overlapping but not identical patients depending upon exactly the data that were available. And one key point is that we concentrated on the change in hemoglobin levels from the initiation of treatment out to week 4. And that was what we focused on for the genetic analysis. And I want to just make one comment about how you carry out one of these genetic studies. You really cannot face the full onslaught of clinical complexity normally when you carry out one of these studies. You really have to zero in on some relevant part of all of that data, or there's really nothing that can be studied genetically. And part of the art of doing one of these studies is figuring out what you concentrate on. Here, we decided to look at hemoglobin from initiation to week 4, because at week 4 it's where it got complicated, because doses would be adjusted or maybe ribavirin would be stopped. But from 0 to week 4, not many changes took place, and it was already known that a lot of the change in hemoglobin that would occur would be seen in those first 4 weeks. So that was a way to capture a lot of what we wanted. So that's what we did. We just looked at either the individuals that suffered beyond some threshold decline in hemoglobin, or we looked at a quantitative reduction in hemoglobin from baseline and carried out a genome-wide association study. And we got identical results with both of those formulations for the hemoglobin change. And what we found this time is a set of variants on chromosome 20 that, again, show clear genome-wide significance. Now, for those of you for whom it's getting a little bit routine to see one of these plots that suddenly get a bunch of these red points that go off into the stratosphere, let me just ask you to talk to anybody that works on the neuropsychiatry side of the center how often this happens. This really doesn't happen that often when you do a genome-wide association study, that you get effects like that. So these really are standout effects. But here, what we are seeing is a set of points that go up, in this case, near 10 to the negative 50. So again, absolutely no question, we have a real association with who suffers the most severe decline in hemoglobin. Here are some of the p-values. Before I do that, so there are the variants that are associated in the genome-wide association study, and all of those variants cluster in this open reading frame right here. Okay? So here are the variants that show strongest association. There are the p-values up towards 10 to the negative 50, and there's the open reading frame in which the variants reside. However, that open reading frame whatever that open reading frame is, that open reading frame has nothing whatsoever to do with who suffers anemia and who doesn't. It turns out we know what's happening is that these variants right here sitting in this open reading frame are marking the effect of functional variants in this gene. Okay? Now, the way that we worked that out is that we had these variants, the discovery variants, and then we looked in the region around those and asked, are there any known functional variants that might be responsible for the effect? And we immediately saw, in fact, the same day, that there were two functional variants in this gene, ITPA, okay? So that encodes an enzyme that breaks down inosine triphosphate. I can tell you more about that in a minute. Two variants that were in this gene that were both known to be functional and known to reduce the activity of the enzyme that that gene encodes. So we got interested in that gene, and what we did is we looked at the p-value for those two functional variants when we genotyped them in the full cohort. So those functional variants were not on the chip directly, but what we were hypothesizing is that their effects were being visualized on the chip by these marker variants that were in a gene a few genes away. Okay? I'm not going to go into why that happens. It's a well-known property of association that you can get these indirect associations. In fact, that's the whole idea of taking a subset of variants that represent other variants. So that's what we thought was happening. We genotyped these variants in the full cohort, and what we found is that they had very strong association. In fact, when you looked at their independent p-value, so looking at the p-value for this variant, controlling for that one, and vice versa, the p-value is even stronger for one of these functional variants than the discovery p-value of 10 to the negative 50. So that made us think maybe we really have discovered the cause of this association. And so what we then did is ask the statistical question, do we actually have the cause of the association? And you can ask that statistical question in the very obvious way. You construct a regression model taking the discovery variant, okay, and you put these two functional variants into the model, and you ask, does the signal that you initially reported go away? So has it been explained by these two functional variants? And that's what happens here. So in both individuals of European ancestry and African ancestry, there are the discovery p-values for what we discovered in the initial study, and here's the adjusted p-value when you put those functional variants in. Look at what happens in European Americans. The p-value goes from 10 to the negative 45 for the discovery variant to 0.9. The effect completely goes away. In a statistical sense, this is an explained genome-wide association study. Okay? And for those of you who have been following the, that was definitely Freudian, following the discussions that I have had with Francis Collins over the years about genome-wide association, it is not about whether real things are being found. Real things are most certainly being found. The difficulty with genome-wide association is that it's not that often that you track down from the association signal to the underlying cause of the association signal. And when you don't track to the underlying cause of the association signal, it's difficult to use the discovery for predictive value, and it's difficult to gain the biological insight. That's what the discussion is about. Not that it never happens in GWAS, but it rarely happens in genome-wide association. And from the hundreds, literally now hundreds, of significant associations that have been reported using genome-wide association approaches, this is one of a handful that have been tracked down to cause. Okay, so that is, in fact, precisely why I am so much more bullish on sequencing approaches, which you will hear about soon, than GWAS. Not that GWAS doesn't work. It's that it doesn't give you the cause often enough. In any case, this time it did give us the cause. This is just an illustration of the clinical importance in this case. One way to look at that is by looking at individuals broken up by genotype in terms of their predicted ITPA deficiency. So just look down here at the final outcome. What I do is look at the predicted activity of this enzyme based upon the genotype state of the two functional variants that reduce enzymatic activity. And then I break patients up into those that have no reduction in enzymatic activity predicted or maximal reduction in enzymatic activity predicted. And then I look at individuals that either have one or another measure of severe hemoglobin decline. I won't even bother telling you what those are. It doesn't really matter at this stage. And ask what the proportion is in these different groupings of patients. And what you see, individuals that have no deficiency for this enzyme, they are more than half of them will suffer one or the other, which are overlapping measures, significant decline in hemoglobin levels. And this is a decline that could trigger changes in dosing of ribavirin. Whereas individuals that have maximal predicted deficiency in this enzyme's activity, none of them have that degree of reduction in hemoglobin. So what we have here is a genotype that appears to be completely protective against one of the leading adverse events associated with this therapy. Now, one question here is what might be the clinical relevance, okay? One clinical relevance I think that you can largely rule out is getting away from having to monitor patients. You're still going to monitor patients for a whole bunch of reasons. So I don't think that it changes much in terms of knowing that a patient is not likely, if you're going to initiate treatment, to suffer hemoglobin decline. But where there is, I think, clinical relevance is that some patients are contraindicated from this therapy precisely because of concerns about reduction in hemoglobin. And if those individuals are of this genetic makeup, they can still be treated because you know they're not going to suffer significant hemoglobin decline. Okay, so I also said that this one was a bit more encouraging in terms of mechanism. It turns out, thanks largely, or in fact entirely, to the work of Yuki Hitomi, who is a new postdoctoral fellow in the center, we actually believe we understand exactly how this is working biochemically. So I don't want to go into much details here, but just to say this is a part of what we believe is the full path of discovery genetics, which is trying to understand the underlying biology involved. So what is now recently thought to be at least a primary driver of ribavirin's effect on hemolytic anemia is that ribavirin reduces GTP levels. And the reduction in GTP levels can lead to a reduction in ATP levels, which are required for cellular homeostasis, and so you can get an increased propensity to lysis if you have a reduction in ATP levels. The enzyme that the variants are located in, ITPase, breaks down ITP, as I said before, and it's already known that if you have deficiency for that enzyme, you get a buildup of ITP in red blood cells. And so the hypothesis was that that buildup of ITP in red blood cells could somehow help to maintain cellular ATP levels despite ribavirin's effect. And it turns out that that is exactly what happens because it's known that this first committed step for ATP biosynthesis is catalyzed by an enzyme that requires GTP, an enzyme called ADSS. So you get a reduction in GTP, you get less of this committed step and a reduction in ATP. It turns out that ITP is able to stand in for GTP and to maintain that catalytic step. And we were able to show that in vitro. We were also able to prove that that's what's happening in vivo. And the way that we could prove that that's what's happening in vivo is we took red blood cells from individuals with different genotypes, either having the protective variants or not having the protective variants. We challenged those red blood cells with ribavirin, and we looked at the change in ATP levels over time. And what we see is that individuals that are protected genetically have less of a reduction in ATP levels than the wild-type individuals that are not protected. But most importantly, that protective effect is ablated if you use 6-MP, which inhibits that enzyme, which is on that committed step to ATP biosynthesis. So this shows that we've actually traced down to mechanism. Okay, so that has, I hope, really made the case that there are situations in which common genetic variation that's sitting in the human genome has an impact on drug responses that we would care to know about. And I think that that really makes a strong case of committed investigation into the role of common variation for certain drug responses. But it is also the case, very clearly, that there will be some responses to drugs where the variants that are important are too rare to be picked up by genome-wide association. And one way to think about that is that the variants that influence the disease themselves we now know with some confidence will often be rarer than can be picked up in genome-wide association studies. That's what's one of the main things to come out of genome-wide association. And so those will sometimes surely influence the best treatment for a patient. So in that setting, at least, and surely others, we want to have a framework in place to be able to also identify rare genetic contributions to drug responses. That is now also systematically possible thanks to developments in whole genome sequencing. And I'll now just tell you a little bit about those developments and the pipeline that we've established for this in the center. So the first question is, does it work? Can you actually take a genome that's got a variant in it that you're interested in, sequence that genome, and find the variant? So the answer to that question is yes. As I alluded to earlier, we took a family where we knew there was a Mendelian disease there, but there wasn't good enough evidence in the family to find the disease gene by traditional linkage approaches. So we took one member from that family, we sequenced that one individual from the family, and then we looked in the regions of the genome that had a little bit of linkage evidence, and the mutation that is responsible for the Mendelian disease just jumped out, waved hello, and said, hi, I'm the causal mutation. Why don't you write up a paper, okay? So it really does work. You can actually sequence a genome, and you can find the mutations that you're looking for. So that paper is there, and I think it was either the fourth or the fifth time that something like that had been done with whole genome sequencing for Mendelian disease. So what happens when you carry out a sequencing study? One point I really want to emphasize is that you have to be extremely careful with who you decide to sequence because it's expensive and you're going to be trying to make fine distinctions based on genetic variation amongst patients. So you need to be really clear about who you select to sequence. You then do the sequencing. The way that works now is that you align short reads back to a reference genome, and then you call all the variants that emerge, and you get millions of variants for each individual genome that you sequence. And so one of the key things that's required if you want to carry out these kinds of studies is you have to have some framework in place for interpreting all of that genetic variation. So what do you do with it all? Well, what you do with it all is you give it to Dongliang Ge. So what I would say is that if you tried to do this 2 or 3 years ago, you would be more or less dead in the water. And the reason is you'd have all these variants in each of the genomes that you sequenced, and many of them would not be in any variant database, and you wouldn't have anything to do with them. But what Dongliang started developing in the center, in fact now I think starting about 3 years back, is a bioinformatic environment that takes variants that you see in the sequencing, whether they are already known in public databases or they're brand new, seen for the first time in those individuals, and then the bioinformatic environment you set up interrogates public databases to ask the question, well, if I had a variant at those genomic coordinates, what would the variant do? Would it do something to a gene? Would it do something to some other annotated feature of the genome? And his software routines can do that in real time for all the genomes that you sequence. So we actually can look at all the variants, and at least for those variants that do something to an annotated feature of the genome, we can categorize them. Without that, you can't do any of this work. Anyway, this thing is publicly available now, so anyone who's interested can have a look at the website. We've been using it to ask what kinds of genetic variation emerge when you sequence human genomes. A paper by Kim Pilak and Kevin Cheyenne and Dongliang Ge just appeared in PLOS Genetics that characterizes the variation that comes out of sequencing 20 human genomes. I just want to tell you a little bit about what we're seeing in human genomes. What this shows is the number of new single nucleotide variants per genome that are identified that are not known either in the world or to you, okay, as you sequence more genomes. So the first genome you sequence, genome 0 here, you ask how many differences to reference does that individual have at a site that's not recorded as having a polymorphism. And the answer is more than 400,000. And the next genome is asking the question, well, now how many do I see that are not in databases and not in the last person, okay? And after you've sequenced the 15th genome, you still are learning about 100,000 new variants that you didn't know about before, okay? And here's a more striking thing to look at. What this is showing is the same kind of analysis, but asking how many genes are you observing that are knocked out, predicted to be knocked out in the individual that you've sequenced? So here you're looking at protein truncating variants that are homozygote. Okay? And what we say is that in this first individual, we learn about 50 genes that we didn't know about before that are knocked out in the person. And after we get out to this point, we still have 3 to 5 genes that we learn about in every individual that in that individual, the gene product is not being made. Okay? So there's people here that are carrying their own broken genes that the community doesn't know about. That'll always be the case. So what this is teaching us is that human genomes are stuffed full of what are almost certainly mistakes. So there is a low level of pathogenicity of variants floating around in human genomes, certainly reflecting a kind of mutation selection balance. There's a tremendous amount of functional variation in the human genome. One practical complication of all of that variation is that it does mean that if you sequence a patient genome and you find something broken, you most certainly cannot infer that that's the cause of whatever the disease is you're looking at. You have to know that that doesn't happen in controls. Okay, so I now just want to give you a little bit of an idea of what we're doing with these kinds of technologies across the sort of portfolio that we're running at the moment. And then I want to say a little bit about the DCRI Discovery Genomics program. So here's some examples of sequencing studies that are underway. We have studies looking at extremes of control of HIV. So here we look at individuals that become HIV positive and then have a loss of immune function very rapidly, or individuals that become HIV positive and go a very long time without having loss of immune function, and we compare those. We also look at individuals that appear to be completely resistant to infection by HIV. We also have, because we are very stubborn, a continuing commitment to neuropsychiatry, despite how difficult that is. And we have sequencing discovery programs going on in both schizophrenia and epilepsy predisposition. We're also running a series of studies on drug-induced liver injury in partnership both with DILIN, that's a NIH-funded consortium to look at drug-induced liver injury, and also the Severe Adverse Event Consortium. Severe Adverse Event Consortium also, we also have a partnership with them looking at agranulocytosis, which is an adverse reaction that occurs for the most effective antipsychotic, clozapine. And then we've got some other discovery projects underway. Here's a sort of expansion of the pharmacogenetics projects. In addition to the drug-induced liver injury sequencing, we also are looking now in collaboration with the, again, Severe Adverse Event Consortium at the effect of some drugs on cardiac arrhythmias. Okay, so where are we in summary? So in summary, it is now emphatically, clearly possible to screen the entire genome for any variant, rare or common, that has a strong influence on therapeutic response or susceptibility to adverse events. And I would guess that the way this will be deployed going forward is that both genome-wide association approaches and sequencing will be important when studying efficacy. I can imagine applications of both of those areas. If you've got a drug where it looks like there's a quantitative range in efficacy, but no group is really very different in response than other groups, you might try to hit that with the genome-wide association approach first, followed up by sequencing the extremes of the response distribution second. On the other hand, if you had a drug that acted, for example, in some ways like the anti-epileptic drug Keppra, where there is a minority of patients that clearly have a fantastic response, very high seizure burden, Keppra is introduced, dramatic reduction in seizures. In that case, you might sequence the subgroup of exceptional responders. I would also imagine that for rarer adverse events, by and large, the discovery strategy will be sequenced. Look at the individuals that have the rare adverse event and then sequence them. Okay, so what is our capacity right now in the center? So right now, we can process by whole exome approaches about 100 samples a month. Now, what that means is instead of sequencing the entire genome, we use commercially available reagents to capture that part of the genome, or most of it anyway, that actually encodes protein. And then we sequence that part of the genome only. Admittedly, we'll miss some things, but it's considerably right now cheaper to do that and faster because you only have to sequence about 1% of the genome, so it's a lot easier. So right now we can process 100 samples that way, sequencing the entire exome, protein coding part of the genome. And for whole genome, we can do about 30 samples per month in the center. That's the generation of the data and also all the analyses. Where is this headed? Well, here is a graph that we just, in fact, put together this morning to sort of give an idea of what we ought to be thinking about in terms of scale. So how ambitious should we be thinking in terms of how much of this kind of work is actually going to go on across the drug development program in the world as a whole? Well, here is, for comparison, Moore's law for sequencing costs. Okay, so Moore's law is that the cost per number crunch in computing is halved every 18 months or 2 years or something like that. Okay, so if I apply that to the cost per base of sequencing, beginning when next generation sequencing kicked in 2006 to 7, that's what Moore's law looks like. And what does that look like for Kevin Shiana's law for sequencing? Like that. Okay? So over the past year since the introduction of next generation sequencing, the costs are so far past what Moore's law produces, I don't even have an adjective for it. So where is this headed? Well, this is headed towards, we're going to be sequencing in any context where there might be something interesting happening genetically. Now, of course, before long, we're just going to be sequencing everybody in a variety of contexts. But for discovery, we're going to be sequencing when it looks like something interesting is happening. So we need to think about how to do that intelligently. Okay, here's an example of what some projects might actually look like right now. Imagine that you've got a drug that appears to work in some of the patients. You want to look at genome-wide association, let's say, in 1,000 patients. If there was a decision to trigger such a study, the study would take us 3 months to do from the receipt of the DNA to the report of any significant findings, and that study would cost $500,000. So one way to look at this is that if there's something like IL-28B lurking in the genome of individuals that are in some trial for some drug, this is what it costs to find out whether or not it's there, and this is how long it takes. Okay? So there is no question that this is the kind of thing in many settings that you definitely want to know about. What might a sequencing study look like? So let's say that we performed either whole genome or whole exome sequencing on 25 to 50 patients that have a severe adverse event. We compared them to controls that we already have sequenced. Okay, now that's an important point, because if it's a rare adverse event, you actually can sequence just the individuals that have it and compare them to unexposed controls. That'll work okay. A more common adverse event you can't do that for. What does this project look like? Well, if you did it for, whole exome, it cost you about $125,000. If you did it for whole genome, it cost you $750,000. Take you 3 to 5 months. So if you had an adverse event that was important enough to cause a problem for the compound, and you wanted to ask, are there any gene variants that might predict who's going to have it so the compound can go forward, this is what it costs and how long it takes to figure it out. This is brand new stuff. You could never do that before for any variant that might be lurking in the genome. And we also know that there are some variants that have impacts on rare adverse events that are strong enough that they would allow a compound to go forward. A Baskerville hypersensitivity is a well-known example of exactly that. Now you can find whatever they are. Okay, so in recognition of this, folks at the DCRI had the foresight to start thinking about putting together a program so that we could roll this out systematically across the DCRI. Here's just a kind of update of where we are with that. The program officially launches right now. There's a press release. There's going to be a video describing what we're up to and why. And there is a brochure, as you heard from Kevin, outside, waiting outside already. And I should also just say that anybody who's interested in getting more information about that from the center can contact Darcy, who is also here. So that's all I wanted to talk about. I would just like to make sure that I've acknowledge the folks who've been working on the various studies. Many of them are here. They are indicated on this slide. I won't go through and name them all in the interest of time, but I also just want to return to what I indicated at the beginning. For the center, the link up with the DCRI really is mission central for us. We really feel strongly that it is time that systematic genetics and genomics really can make a big difference in the clinic, and it really is a partnership with expertise in clinical research that's required to really make it work. So thank you very much for your attention.
Well, great. Like I said at the beginning, this has been very exciting to put together and to work with David and his team. And actually, I think we started this in the spring and thought about this idea of a launch. So after today, all these tools will be available to everybody to think about for work going forward. Why don't we open this up to questions?
David, that was a great talk. I had a question for you on the hepatitis C. I'd like your opinion on when do you go from association to being able to show that genomically guided strategy actually changes outcomes. So, for instance, with your Hep C work.
Yeah, that is. So let me decide how to answer that. Is Rob here or not? So that's a hard one, and I have my own orientation, and my orientation is that there's two ways that genetics might make genetic or genomic discovery might make its way into the clinic. One way is when there is relative algorithmic certainty, and that I think is unarguable. If you have a situation where, okay, the treatment only works in individuals that have X, and therefore you need to actually test that and only use the treatment if it's present, that's algorithmic certainty. I don't think anyone can argue about that. I think that there's a role for genetics, and I would actually guess that it's going to be the stronger role, to just feed into the clinical gestalt. And that is actually my own orientation, because I think what happens is for many therapeutic areas it's going to remain somewhat of an art form to decide what to do. And when you have strong genetic predictors, it will just influence your thinking to some degree. And so in the case specifically of hepatitis C, part of the thinking is, well, is there liver damage right now? So do I need to treat immediately? Part of the thinking is, what's the likelihood that I would actually cure the infection? Should I wait for newer treatments? That is the reality of clinical decision making in chronic hep C infection today. And genetics is relevant to that. Not that it tells you do or don't algorithmically with certainty, but it's part of that gestalt evaluation. I think that genetics is going to have to be accepted as being used in that kind of a way. That's my own view.
Let me maybe switch just a little bit. You know, one of the issues and one of the reasons for developing the center within DCRI is to build this collaboration between the groups. The thought process in terms of the pricing model is if you're an investigator and want to have access to this technology, the pricing models are all available at the standard. In terms of SOPs, really the issue at the beginning of the study is to make sure that you have samples available on all patients so that at the end of the trial you have material available to bring to David. On the faculty-to-faculty side, how do you think about and when should people think about forming hypotheses about genetic variation in clinical response?
So sometimes I think there's a stronger case than other times. Like the case for hepatitis C, for there likely being something genetic happening, was always overwhelming because we had all these pointers that there was something genetic happening. And the potential relevance was always pretty clear. So I think there will be settings like that. One example would be an adverse event that is rare and that is serious. Another example would be a case where there is a minority of patients with very, very clear outlier responses to treatment. Those would be, I think, unambiguous cases for triggering genetic investigation. But I would also probably suggest that we don't rule anything out, because in situations where we have a quantitative distribution of responses, that's no reason to believe that there's no genetic control there. And so, I really would say that although there are clear cases for where it would be almost malfeasance not to investigate the genetics. I really don't think there are any realistic settings where we can say there's not likely to be a genetic contribution. And now, because it is relatively straightforward, relatively inexpensive, a really step increase in the amount to which genetics is deployed is appropriate.
You might have noticed a lot of the publications were literally the last 18 months that you showed. I think the technology you're talking about today and your capacity to use the tools really are unique or almost unique in the U.S. At least we could say that. What's changed to make this now different than what we've been doing for the last 10 years?
I think probably the biggest change is the sequencing capacity, because genome-wide association has been around for a few years and has not been systematically applied in pharmacogenetics, and I think it has yet a while to run in discovering new things in genome-wide approaches for drug responses. I think the main difference is the sequencing, because what we now can do is really find whatever is there, and that is what is different. And so we actually really can investigate the hypothesis that the inherited genome is having something to do with the responses to treatment that one is seeing. And I think there's every reason to believe that there will very frequently be a strong dependence on the inherited genome. Exactly how often, I don't know. In what therapeutic areas is the most important, again, I don't know. But for any drugs that work, so if we actually take the whole class, take drugs that don't work that well, those are, I would say, a much less high priority for genetics. So if they don't work very well, then there may not be a really good foothold. But drugs that clearly work in some individuals, those give a clear foothold for genetics. And I think what's new is the ability to find whatever is there. I think that's the key.
One of the things that you'll learn coming over to our world is most of the trials that we work on, the drugs fail. They'll fail due to toxicity, they'll fail early, they'll fail late. Really, frankly, on the industry side, one of the things that we've talked a lot about is going back to look at salvage. If I've done some work and it's got some toxicity with a small proportion of people, that doesn't mean you can't do some of the responsible molecules. We don't like to think about failure all that much, but most of our work is dealing with failure. How is this going to work when something doesn't work?
I mean, the nice part about the salvage model is that it is risk-free, too, to a company, in the sense that there is some concern about efficacy predictors that might reduce the market share by telling you what patients not to use the treatment in. But for a compound that you're in trouble with because of safety, the genetics can only help. So.
We're very excited about this opportunity for the faculty to collaborate with you and your center. Industry sponsors, as you might imagine, are not, let's say, the most progressive group of thinkers in terms of applying new technologies that might limit the population that's available to sell things to. What's the countervailing message to that? I said the price tag for $500,000 doesn't potentially sound like a lot in the terms of a $500 million drug development program. But in fact, for the individual investigator who's exploring a hypothesis, that is a lot. How do you balance those two of getting the industry to try to invest in this at the same time allowing a faculty member to explore his or her thinking?
I think that if there's only one major drug on the market that the company owns, it's a hard sell. Okay, that's my honest opinion right now. If there are multiple drugs on the market with different modes of action, I think there is a very clear argument, which is that you might establish market differentiation by showing the patients in whom your drug works better. And so I think that it's a really much easier case to say that you don't need to worry quite so much if there are multiple drugs on the market. But if it's the only one, I think that there is concern that's a little bit tougher, at least for the way that I look at it, to get around. The only other comment you might make is that for commonly used drugs, it's probably the case that it's going to happen for your drug eventually, no matter what. And so there is an argument that you would rather do it than have it done to you in terms of finding out what's there in the inherited genome. But I do think that it's an easier sell to not worry so much about market segmentation when there are multiple drugs on the market.
Well, if there's no other question, let me ask another one then. FDA regulatory policy on this, have you had discussions? What you made your comment about it's going to be done to you, that usually implies that some big brother type entity is going to do that.
So right now, my impression of the way the FDA is viewing this whole area is that they will step in on the safety side, but not on the efficacy side for drugs that are already on the market. They would consider an approach from the sponsor for a marker that might be used as a companion for efficacy, but they won't require that. But on the safety side, they might. And there are contexts in which they would construe an efficacy marker as affecting safety, but it would have to be construed as such for them to step in. That's as I see it right now.
Certainly you're looking at one facet of the genome as well as the expression of the genome, making a living biological system. How do you see your group and DCRI interacting with other groups? Because you talked previously about the fact is we don't want to just know we have a signal. We want to know what it means. So you may decide initially to look at the exome, but there's microRNAs, there's other players out there that we want to look at. So how can we facilitate looking at a problem holistically and thinking at DCRI how we might get the groups together to think it through?
Yeah, that's a great question. So the first answer is that if it's in the inherited genome, it's pretty easy to tackle it in the confines of what we do in the center. So, for example, microRNAs, any variant that's sitting in a microRNA is completely captured by the whole genome sequencing approach, and you could interrogate the microRNAs directly as an annotated feature of the genome. So that's very easy. And that's true for any specific annotated feature of the inherited genome. As we move on to other kinds of genomic things that one might be interested in, then we are talking about situations where we would want to partner with other entities. So metabolomics would come immediately to mind, where there is established expertise, for example, in Chris Newgard's operation, and also proteomics and other approaches like that. So in terms of the discovery side, one can imagine in some context partnerships being established, but for the inherited genome, the center has capacity for whatever kinds of questions emerge. Downstream, for biological interpretation, it's impossible to say anything general because it depends entirely on what you discover. So, depending on what you discover, you might find yourself in the realm of, neuronal electrophysiology or something as bad as that. Or you might find yourself in the realm of something that you can actually do biochemically, like the ITPA in the lab. So it just depends on what comes out.
Yeah, I don't think that we were thinking about this as a terminal destination for this work at all. But I think the ability to actually bring a data collection mechanism to bear within the context of trials could be used for all these other platforms as we continue to progress. So, so I think that's a great opportunity. But I think from our side and from dealing with their sponsors, what we are offering is a way to think about this, a way to make sure the samples are stored appropriately with the right kind of barcodes on them in an accessible way. And to have all of this will interface our EDC platform will interface with David's sample ID numbers so that we will be able to link the things up and there won't be quality assurance problems down the road. So that kind of really exciting infrastructure can be repurposed to other areas, but that's what we'll have the capacity to do going forward. Well, David, thanks a lot for coming over here, and thanks everyone for joining us today.